Good day, everybody. Welcome to exercise 23 and 25. We're going to be learning about the anatomy of the eyes and the ears. We are in the home stretch here. These are the last two exercises for this AP1 lab. So let's get started with the anatomy of the eyes. We have this illustration here to show you the outside structures of the eyes. The eyes is protected by three layers on the outside. Starting off from the outer layers, we have the sclera here, and the sclera here is just the connective tissue protective layers, and the middle part is called the choroid, and this is filled with a number of blood vessels feeding nutrients into a number of cells inside of our eyes. The inner layers here is what we call the retina, and the retina is the layers where we take the photo, the light information, energy, and turn it into neuronal signal that your brain can understand. And we're going to turn this retina here, which is lined with a number of photoreceptor. We'll talk about a photoreceptor in just a little bit, and send it out through the optic nerves to the brain. Now coming toward the front side, we have the sclera that continue toward the front and turn into a transparent layers called the cornea. The reason it has to be transparent so that the light can pass through the cornea into the space here, the filled with fluid called the aqueous humor. Right behind this aqueous humor is the layer called the iris. This is where we have our eye color. This example here, in this illustration is a blue eye. In the middle of the iris, we have an opening called the pupil that allow lights to pass through and going into the lens. The lens is how we focus the focal of the light so that it ends perfectly in the area of the retina, in the area we have the fovea. I'm not going to go into the function of fovea, or keep it simple. This is where we have the sharpest picture in our eye. Gonna move on to the next couple slides, going into more structures of the eyes that they do not have it here on this illustration. So I'm gonna skip to a couple slides here talking more about what's going on that hold up the lens together. So the lens here is not just floating inside the eyeball, it's actually suspended by the suspensory ligaments and can be shaped more into more flat lens or more of the obtuse lens. It's kind of rounded lens. This is thanks to the ciliary body or ciliary muscles is what help adjust the shape of the lens to make it more round or make it more flat. Allow us to adjust the focal of the light. Behind the lens is another cavities that's filled with another group of fluid. These fluid here is what we call vitreous humor. Allow us to maintain the pressure of the eyes. One thing about the lights and how we focus the light is the pupil. And the pupil here can be adjusted as well. You can make the pupil larger or smaller. What makes the pupil smaller is the sphincter pupillae muscles. This muscle will activate. Often happen when you're looking at something bright. The opposite of that is the dilator pupillae muscles, is when you dilate a pupil to allow more light to get into the eyeball so we can see better in the dark. Continue with the inner layers of the eyes. We have the melanocyte to help absorb the lights and the vitamin A to help us renew the photoreceptors. In addition to this photoreceptor, make sure you know the two types of the photoreceptors. We have the rods and the cones. The rods photoreceptors help us process gray tones. So things like the lighter color, the darker color works really well in the dim light in the dark rooms. Cone photoreceptors help us process the primary colors, help us see things in colors. The thing about the visual information and visual processing is that the photoreceptor kind of hiding all the way to the back of the eyeball. So the light has to go through other cells in order to get to the photoreceptor first, and then the photoreceptor pass it backward toward other cell, the bipolar, and then to the ganglion cell. Once the ganglion cell receive the neuronal signal, they bundle together into optic nerves, pass out to the back of the eyeball through the opening called the optic disc. We show you the photoreceptor, the bipolar cell, and the ganglion cell, and then we'll get to see the optic disc and optic nerves. So have a look at the next slide here. We have this photoreceptors, and the light has to come through this area here, which by the way, this is where we have the lens of the eyes, and just kind of show you that we have the eyeball here. As the lights pass through the lens, pass through the vitreous humor, it has to go through the ganglion cell, the bipolar cell, in order to get to the photoreceptor, the rods and the cones. It sounds counterintuitive, right? Why are we not having the photoreceptor toward the front side, right? So once the photoreceptor, the rods and the cones receive the signal, they transduce the signal into the electrical signal, the action potential, things that neuron can understand. Pass that information with the dendrite of the bipolar cell sends signal to the axon, which are these area over here, to the ganglion cells. The axon and the ganglion cell converge together, bundle together into the optic nerves, and the optic nerves comes out from the eyeball on the back side. So here is the opening, it's the optic disc, and we have the optic nerves that comes out from this area here. We'll review everything that we just covered again when we get to the lab together. We get to see everything's again on the actual models. Before we get to that, however, let's continue with the extraocular muscle first. Just kind of review the names, the cranial nerves that innervates them, and then we'll get to review that again when we get to the lab so that we have one video all together. The extraocular muscles surrounding the eyeball allow us to move our eyes in different directions. There are 6 different extraocular muscles innervated by 3 different cranial nerves. So start off with the top part. We have the superior rectus, inferior rectus, medial rectus, and the lateral rectus, which by the way, this is a right eye. And the last 2 muscles are the superior oblique and the inferior oblique. The thing about these muscles, the extraocular muscles that are innervated by the cranial nerve number 3, the extraocular nerves, are the superior rectus, the inferior rectus, medial rectus, and the inferior oblique. I kind of drew a wrong arrow, so make sure one more time. Superior rectus, inferior rectus, medial rectus, and the inferior oblique, innervated by cranial nerve number 3, the extraocular nerves, which by the way, I have it on the table here to the side as well for your reminder. We also have the superior oblique, innervated by the cranial nerve number 4, called the trochlear nerves. And the last one, we have the lateral rectus, innervated by the cranial nerve number 6. So by the way, the cranial nerve number 6 is like this. So we have a V followed by the one line, whereas the superior oblique is innervated by cranial nerve number 4, which is CN, and the line is in front of the V. So here's the cranial nerve number 4 for those of you who needs a little bit of help with the Roman numeral. And the function of these guys here, I have it off to the side. I will have a review when it gets to the lab as well. We have an eye model here. So let's have a look at the structure on the outside and then we'll work our way in. So start off from the outside here, we have the white tissue. It's what we call the sclera. And right next to that, we have another gland here called the lacrimal glands. It's basically your tear duct. So when people cry, the tear comes out from this guys here. On the back side here in red color, these are your extraocular muscles here off to the side and off to the side. We'll come back and revisit these guys here another time. Let's have a look at the inside here. I'm going to open this up. Let's have a look at the inside structure. Start off from the external structure. Let's have a look at this white part over here. This is the sclera that we talked about earlier. It goes all the way around the eyeball. In the middle layer here, in brown color, this is called a choroid. It has a number of blood vessels that feeding into a number of cells inside the eyeball. We also have the inner layer here called retina in the yellow-orange color here. It looks like an egg yolk color. This is where we have a number of photoreceptors that allow us to process light. We'll come back to the photoreceptor in just a little bit. Now let's continue forward. We have this area here in the front. I'm going to take this structure here and kind of show it to you that we have the transparent area here. It's called a cornea. Inside of this area here, we have the brown structure. It's called the iris. This is where we have the eye color. So this example here would be someone who has the brown color eyes. In the center of the iris, we have a hole in the center. It's called a pupil. This is what allows light to pass through and going into the inner part of the eyeball. Now, let's have a look at the other half of this model here. We have this model, and right behind the iris, we have this structure here called the lens. What holds the lens together is the suspensory ligaments, which we do not have here on this model. Instead, on this model, we have the ciliary muscle. It's also supposed to attach to the lens. This ciliary muscle is what allows us to adjust the shape of the lens, allow us to focus objects, whether it's the object is closer to the eye or further far in the distance. It's what allows us to focus the light so that it fall right into the exact spot where you have this photoreceptor. We'll get to that in just a little bit. Now, let's have this put together real quick so we can get to see the other structure together. Notice that we have two bubbles here. We have this kind of plastic ball looking thing here and the space in between your cornea and the iris. The space inside of this area here is filled with fluid called the aqueous humor. The space in this area here in the middle of the eyeball is filled with fluid called the vitreous humor. And another thing before we move on to the structure here is that we also have the muscle and the pupil in the iris. And let's have a look at the iris one more time, and then I'll put it back. That notice that you got a pupil here. Now that we see the back side, we'll see it better that this hole is called a pupil. And here's the back side of the iris. We have muscle called the sphincter. Pupillary muscle is what allows us to constrict the pupil, allow less of the lights to pass through. This happens when you look at something that's nice and bright, like say you walk outside and you're looking into the bright light. The other muscles do the opposite. So instead of constrict the pupil, there's a dilator, pupillary muscle, that allows us to dilate the pupil, allow more lights to pass through the pupil. This happens when you look at the dim light or you walk into the dark rooms. Let's put this back here. And as the lights pass through, so I have the laser pointer here represent the lights. As the light pass through the cornea, through the pupil, through the lens, it continue all the way to the back side called the retina. So it's the retina that we talked about earlier, continue to the back side. This retina here, this layer is filled with these cells over here represented by the blue, by the red, and here in the green color. Start off with the blue cells first. So we have lights that pass all the way to the ends, to the back side of the eyeball. These are the rods and the cones photoreceptors. These photoreceptors process the lights and turn it into electrical signal called action potential. Pass that signal to these cells over here, to these cells called the bipolar. We have the dendrite, cell body, and the axon. So I got another, let's use another structure here. I have a little bit of reflections. We have the dendrite, cell body, and the axons. Hence why we call these cells here in red color the bipolar. They send a signal to these ganglion cells. And notice the ganglion cell here, the axons of the ganglion cells converge together. And what comes out off to the back end of this one here. So you can notice we got a hole in the back side, hole in the back side. Here's the other half. And if I were to put them together, we have this hole here on the back side of the eyeball. This is what we call, when we align them together, this is what we call the optic disc. It's basically the opening that allow the optic nerve, which is the cranial nerve number 2, to come out. We don't have the optic nerve on this model here, but I can show you on the other model. We're gonna move this to the side and gonna have another model here. Here's another model. We're gonna have a look at the muscle in just a little bit. Here's the back of the eyeball optic disc. And what comes out of the optic disc is the optic nerve, cranial nerve number 2, has a function of carry sensory information, visual information toward the brain. Now let's have a look at the muscles of the eye. We have 6 different muscles of the eye, so I'm gonna do a little bit of MacGyver. Turn this over here. I'm gonna use this as a base so I can put this toward the top so you can see it much better, right? So, all right. Let's have a look at the muscles here together. We have 6 different muscles. We have 4 that kind of in the north, south, east, and west side of the eye. So we have the superior rectus, medial rectus, inferior rectus, and the lateral rectus. We have the other muscles kind of off to the side, kind of on the oblique side. We have the superior oblique and the inferior oblique on the inner side. So I'm going to turn this so they can see the 2 muscles. There are 3 cranial nerves that innervates the extraocular muscles, like these muscles here. Start off with the cranial nerve number 3. The cranial nerve number 3, the extraocular nerve, innervates the superior rectus, medial rectus, inferior rectus, and the inferior oblique down here. So 4 muscles innervated by the cranial nerve number 3. The lateral rectus here, off to the side, is innervated by cranial nerve number 6 called abducens. And the last one here, the superior rectus, I'm going to turn to the side so you can see superior rectus. This is a tendon of superior rectus. This muscle here is innervated by cranial nerve number 4, the trochlear nerve. And I'm going to have a cutout. I just want to show you how we have a look at this extraocular muscles and the function of these guys here. So this is just for the innervations of the extraocular muscles. So I'm going to cut you to the other side of this one. So I cut to this face here. It's a nice and beautiful drawing of a face. And we'll focus on the eyes of this face over here. We'll focus on the right side, this one over here, because I can only do one side at a time. And I have this model here to show you the 6 different extraocular muscles. We'll start from the top here, this superior rectus. This is what allows the eyes to look up, straight up. This inferior rectus down here is what allows the eyes to look down, straight down. The medial rectus here off to the nose side allow the eyes to move toward the nose, the medial rectus. And here the lateral rectus is what allow the eyes to look to the side. Now the other 2 oblique, we have the superior oblique here. This is what allow the eyes to look down and out, just like this. There's another muscles, the inferior oblique is what allow the eyes to look up and out. So these are the function of 6 different extraocular muscles. Welcome back. We're gonna finish up this exercise with the structure of the ears. The ear is pretty straightforward. So we start off from the outside. We have the pinna, or the auricle, or the ears, the outer part of the ears, and they have a function of gather all the sounds together and transmit it through the ear canal. And we also have very unique tissue called elastic cartilages that maintain the shape of the ears. It is so good that when someone were to say twist your ear and let go of it, your ears bounce back. So that's a unique characteristic of the elastic cartilages. Once the sound is transmitted into the ear canal, it will vibrate the eardrum or the tympanic membrane. The vibrating tympanic membrane will transmit the vibration to the 3 bones of the middle ears, which includes the malleus, the incus, and the stapes. The easy way to remember the names of these guys here, the malleus, incus, and stapes, is that they are M-I-S. They are the miss. And once the stapes vibrate, it will also transmit the vibration to the oval window, which is the connections between the stapes, this guys right here, to the vestibules. The vestibules connects to the semicircular canals. There's 3 semicircles loops that allow us to maintain the orientations of our head, is sensing the orientation of our heads, whether your head is tilting upward, downward, tilt to the side, or turn side to side. That's how we know our head is moving in which directions. Next to the vestibule, we also have the cochlea. It kind of looks like a shell of a snail. And this cochlea here is lined with a number of stereocilia on the inside. When there's a vibration, it will send the frequency, and the vibration will happen in different area of the cochlea. That's how we know the different frequencies of the sounds. And the cochlea will turn those energy into the electrical signal that our brain can understand. And the cochlear and the semicircular canal transmit the vestibule, the equilibrium and the sense of balance, as well as the auditory information from the cochlea to the brain with the help of the two nerves, which are the vestibular nerves and the cochlear nerve. They didn't label here for us, but don't worry about the details of which one is what. On exam, I'll keep it simple and straightforward. Make sure you know the name of the two nerves here, which is what we call the vestibulocochlear. Together, we call them the cranial nerve number 8. So here's CN 1, 2, and 3. For those of you who needs a little bit of help with the Roman numeral, that's number 8. So it's the V with 3 lines on the back side. Cranial nerve number 8 transmit the auditory information as well as the equilibrium to the brain. The next slide here giving you the summary of what we just talked about. And make sure you know the slide here. The next one here is what you do not need to know for the exams. I have a label here that you don't need to know that, but in case you're curious about how the cochlear able to turn the sound wave into action potential electrical signal, you're welcome to read the slide here to satisfy your curiosity. Otherwise, I'll send you to the lab one last time, and here we go. We have an ear model here, so we can review the ear structures that we just went over together. Let's start off from the outside here. We have the pinna or the auricle made out of the elastic cartilages, so that when you twist your ear, it will let go of it, it will bounce right back because of elastic cartilages. Let's have a look at the cross section. It's going to remove this area here so you can see the cross section. On the inside, we have the ear canal, and any sound that comes to the ear, the pinna or the auricle will collect the sounds and vibration, pass it to the ear canal, and will vibrate the eardrums or tympanic membrane. This tympanic membrane here going to remove this structure so you can see the inside. It attached to the 3 bones. Let's have a closer look at this area here. So we have the tympanic membrane, aka the eardrums, that attached to the malleus. Malleus is attached to the incus, and the incus is attached to the stapes. We have malleus, incus, stapes. Easy way to remember these 3 bones here are the M, I, S. The vibration from the stapes will transmit to the vestibule. The vestibule is attached to the 3 semicircular canal. We have the one that kind of goes vertically, horizontally, and off to the side. These 3 semicircular canals is what allow us to sense our head position. If you were to tilt out your head upward or look down, whether you turn your head to the left or the right, or tilt off to the side, is what allow us to maintain our equilibrium and sense our head positions. Connects to the vestibule here is the cochlea. This cochlea here is what transduce the frequency, the vibrations coming from the stapes, and turn it into neuronal signals that our brain can understand. So when we talk about the stereocilia in the lecture earlier, all the stereocilia is inside of this structure here. Notice the 2 nerves that comes out from these 2 structures. We have the vestibular nerve and the cochlear nerve, and together we call them vestibulocochlear nerve, aka cranial nerve number 8, transmitting auditory information and equilibrium information. The balance allow us to balance our body. Now let's have a look at the structure on the inner ear. So we have a cavity right behind the eardrums over here. So when people have ear infections, the otitis media in kids, that's where they have this inflammation and infections. Notice that you have a structure here called the pharyngotympanic tubes. It's basically a canal that allow us to equalize the pressure from the inside of our inner ear to the outside of the environment. So oftentimes when people have to pop their ears when they take a flight, for example, that's where you have this equilibrium, so connection between the 2. All right, that's all I have. The ear is pretty easy and straightforward. And thanks everybody for watching all the way to the ends. Have a wonderful break.